One of the things that I talk to principal investigators and researcher teams at the beginning of a study or when they seek quality assurance and quality control information is about what they've done beforehand, before they've brought their project to us. Have there been different kinds of models that they've created? What kind of preclinical work have they completed already? Do they have standards, and what kind of reagents are they using? There's a wide variety of reagents out there, especially for medical device and for chemical types of trials. And some of them are GMP grade, some of them are research grade, some of them are not for human grade. And it's important to know what we're working with and what the end goal of the principal investigator is going to be in order to make sure that we use the right components in the right parts of the process to get them the result they're looking for. A lot of times research grade material and not for human material, chemicals and whatnot, are not produced in the same types of factories or in the same environment. In addition, when you look at a GMP grade material or something that's for a clinical application, oftentimes it's going to come with a certificate of analysis, a certificate of conformance, and documentation that they've got a quality system in place and that the material is produced in a manner that ensures their safety, effectivity, and potency for the chemical involved. Oftentimes when I talk with PIs and research teams, they're very interested in going in a stepwise fashion. That's been their experience, and their educational background has been to coordinate their studies in a very stepwise approach. It's important when I come in and when the research team and the GMP staff that I work with come in, we look at the project in a broad scope. We think about the entire system all the way from the very beginning to the very end, and then we delve into each individual part. We look at whether they need to create their own solution based on different chemicals or different reagents that they're mixing for the process, and we'll isolate those individual processes and make, for example, a solution preparation record. There may also be cases where they have to do a procedure or a process in a clean room environment, in a biosafety cabinet, and so we'll isolate that as well. If you look at an individual project, it's actually, for us, it's composed of a lot of different individual steps. That makes it easy for us to isolate each individual step, and if we need to make a change, we can make it to that one individual procedure or one individual documentation system, as opposed to changing the entire project that the PI has brought forward for us. Just like a lot of other manufacturing environments or industries, there's a validation life cycle that goes on in the GMP realm. And when we talk about validation life cycle, we talk about the calibration for pieces of equipment, but we also talk about the other 3 or 4 parts of a validation, a qualification that allows a piece of equipment or a process to be used in a good manufacturing process. For us, we start off with a design of qualification, kind of look at the overall process to make sure that our documentation system is going to give us a good set of safety and a good reliability factor at the end to make sure we're doing what we said we were going to do. Then we divide, for example, the piece of equipment into an installation qualification, an operational qualification, and a performance qualification. And each one of those has different steps in the process. The installation qualification may make sure that we've got the right facilities and the right environment for the piece of equipment. The operational component talks specifically about whether the machine, the piece of equipment, or in some cases, a process, is applicable and gives you the result that you're looking for. The last phase, and this isn't always applicable in some certain kinds of studies, but for some processes or some projects that the FDA is very concerned about, they like to see a performance qualification. In that case, we'll actually perform a test where we'll challenge the piece of equipment or the process to its extremes to make sure that we've got a good, robust system. So that entire process, the calibration, the installation, the operation, and the performance qualification put together comprise a complete qualification of a piece of equipment. The same types of processes apply if you're looking at a process, whether it's an aseptic process or whether it's a cleaning process. You want to break it up and look at an I, O, and in some cases a PQ. The FDA is really looking at things in a risk-based perspective. They want to understand the process, but they also want to understand that you know the risks and the potential complications when you bring a project forward. Oftentimes in early investigation when you're doing preclinical work, oftentimes they're not going to be as stringent and demanding on certain aspects, as long as you're able to justify it and make sure that you show that you've thought about it and you put controls into place. For example, you may not be able to use a GMP grade reagent because one doesn't exist or one's not been created yet. But if you use that research grade component and you document all of the systems around it, that you've got it from a good vendor that's been qualified and that you've used have a lot of data beforehand to prove that it's safe and effective, that's one of the things that you can make a risk-based decision on and prove and show to the FDA, hey, we've thought about this, we've done the analysis, the risk on this is low, in our opinion, and we're going to continue moving forward. So that's one of the ways, if you run into a sticky situation or you're thinking about using something, using a system or using a reagent that may not always be FDA approved or granted, you want to think about it in a risk-based model, and that will definitely help you and help them understand your thinking about the process. A lot of times we'll look and see if there's an equivalent product out on the market or something that's similar, that's maybe a little bit different, but the results are going to be the same. We can kind of look and see what they're using. When you're dealing with early projects and the FDA, they're very concerned with safety. Later on down the road, they'll be more concerned with efficacy, but safety is the big thing. So we look to make sure that their sterility, their endotoxin, their mycoplasma, and even something as simple as a gram stain have been brought into the process, or they've thought about that. These are really well-characterized tests that they've been doing for a lot of different projects. And so when we talk with the principal investigator, we make sure that they have an understanding about where that's going to fit into their system. Oftentimes we'll find that we've got to add additional steps or additional checks when they're doing something to make sure that they're doing sterility checks all along the way. Sometimes they say, well, I want to do mycoplasma or sterility tests every time. That may not necessarily be the most advantageous. It takes time to do these tests, and if there's really no benefit to getting the data, sometimes we can sit down with them and figure out what's the best, most effective schedule for them to do the tests. They don't need to do the tests every single time, but if there are key parts where there's personal involvement, or if there's an open container issue, or they're doing some sort of manufacturing process, that may be a time where you want to evaluate sterility and endotoxin, mycoplasma, or gram stain. I think having a good understanding of what the responsibilities and what the expectations are when they work with the CRO is very, very important. Knowing what deadlines and the timeline that the PI is on is also going to be important. It's really important to engage early with the CRO to understand not only the timelines, but what are the roadblocks that the CRO sees in the process. Remember, they've been doing this, they've probably done this for other projects, and so they've got a little bit of experience at this. However, the PI should also keep in mind this is probably a unique process or a unique product that they're bringing to the CRO, so there needs to be a good collaboration and discussions, open communication between them to understand what the best way to get their product through the system and test it effectively at the CRO. Open communication, and the other thing I would also mention is realistic timelines. It's unrealistic to think about a project going through in just a matter of months. This may take 6 months, this may take a year, and being realistic in your expectations and in your conversations with the CRO is critical.